Hi everyone, welcome to the class 4 series of enzymology, and in this series we will be studying about the catalytic mechanisms. So before going detail into different catalytic mechanisms employed by the enzyme, we will be studying what are different types of mechanism. So the general catalytic mechanisms employed by enzymes involve general acid-base catalysis, covalent catalysis, and metal ion catalysis. So acid-base catalysis is based on the transfer of protons. So protons will be transferred either from the enzyme to the substrate or from the substrate to the enzyme, so that the reaction will be facilitated. And the second type of catalysis involves transfer of electrons. So in this type of catalysis, either a electron rich group or electron deficient group will donate their electrons or accept their electrons, creating a transient bond with the substrate, and that later on will be much easier to undergo the further reaction. The third category of catalysis involves metal ion catalysis, in which a metal ion is involved in the activation of some of the groups. So in today's video, we'll be discussing the brief overview of different mechanisms. So first, coming to the different mechanisms, here it is a diagrammatic representation of different types of groups that may participate in a enzymatic reaction. So this is an enzyme active site, and here we have presented different groups. So group 1 is a general acid group which can donate a proton. Group 2 is a general base which can accept a proton. Group 3 is a nucleophile. So nucleophile means it is rich in electron, and it can donate its electron or share its electron to some electron deficient groups. And the fourth one is electrophile. So electrophilic means they love electrons, so they can accept the electrons from the substrates or the intermediates, so that they can be stabilized. And the fifth one is a metal ion. So nearly 1/3 of different enzymes have a metal ion attached to their active site, and this metal ion facilitates the stabilization of a negatively charged intermediate or the generation of a nucleophile, so that nucleophile will be used in further experiment. So what are different amino acids which can donate a proton or accept a proton? So aspartic acid and glutamic acid, as their name suggests, they are acids, so they are already in ionic form at pH 7, where most of the reactions goes on: histidine, lysine, cysteine, tyrosine. So depending on the pH in which the reaction is going on, so they can either accept a proton or donate a proton, so that they can act as a general base or a general acid. So here I want you to remember that these histidine and cysteine, they are mostly involved in transfer of protons, because their ionization is facilitated around the pH 7, because they have the pKa value of 7 approximately. And the enzymes which employ this acid-base catalysis is chymotrypsin, which is a very classic example of well-studied enzyme where this acid-base catalysis is employed. So another enzyme is RNase A. So RNase A has two histidine residues, histidine at 12th position, which act as a general base, and histidine at 119, which act as a general acid. So just as an example, we can analyze how they facilitate a reaction. For example, keto-enol tautomerism is a very common reaction that is employed by many metabolic pathways. So this is the keto form. Here we have the ketone group, and this is the enol form where we have the alcohol group. So this tautomerism is a isomerization reaction where we can see that this group is rearranged. So H from here moves here, and the bonding is, the double bond breaks down. So this is the transition state where the substrate is not yet converted to product. So this is a transient state in the reaction where this H plus needs to be stabilized. So for example, in an acid catalysis, a general acid which is denoted as HA, and this is it is a group, acid group of an enzyme. So this HA can stabilize the negative charge developed in the oxygen of carbonyl group, so that this will facilitate the conversion of the keto form to the enol form. And this H plus, which is donated by this HA group, is again regenerated when this H plus is accepted by this group. Hence, the HA group from the enzyme is regenerated. So here we have to remember that the enzyme never changes. If it is originally protonated, finally it will be protonated and it will facilitate the reaction. Similarly, if it is a general base, the base will accept this proton in the transient state. So it will stabilize this proton and facilitate the rearrangement of different bonds. So this negative charge on O will form the this will upon acceptance of a proton from the surroundings, it will be converted to the enol group. So here the H will again be dissociated from the base with the exchange of some proton accepting groups, and it will lead to the regeneration of the enzyme A. So if the base is in non-protonated form, finally this non-protonated form will be regenerated. So this is a example through which the acid-base catalysis takes place. So in subsequent videos, we will be studying about the mechanism of chymotrypsin, where the histidine 57 acts as a general base to abstract a proton from the serine 195, thus making the serine 195 a powerful nucleophile to attack the peptide bond. So in the subsequent videos, we will be looking at the covalent catalysis.